Um, Great. So thanks everybody for staying to the last end. I'm going to try to go ahead and go quickly around this. So, oops, I don't know how this works. Yeah. So before everything, I would like to acknowledge all the funding that I received from FIU, which was essential, especially for international students. There's less options to actually get funding to support research. So I'm very glad that we have this opportunity here. So I took this picture once over the canopy. So it strikes you a lot because you can see a lot of different plants that look a lot like each other, but it's very different and they actually belong to different species. So this is from the canopy. And when you actually think about the understory, it also happens there. So this is a group of species referred as species swarms in tropical forests, where they belong to groups of plants that do look a lot unlike each other. So they look all green, they have basically the same reproductive structures, they are small, and there's not much difference of how they will recognize them as different species. So among these groups, I'm focusing on the genus Piper. This is the genus of the black pepper. And there's a lot of species coexisting at a single site. So as you can see, they don't differentiate much among the species. They all look green. And actually, they are so underappreciated that even Costa Rican botanists would refer them as to be boring plants that look so alike, not as pretty as many other flowers that you would see around the forest, and they look just the same. But actually, these plants are not the same. They have very different chemistry, and so this this graph is showing you the phylogenetic tree on the left. So you can see here it's facing next to the chemical dendrogram. So this is associating species according to their chemical similarity. And now you can see that, for example, a sister species here like Piper reticulatum and mandarinense, although they are very closely, phylogenetically speaking, they are very closely related. In terms of the chemistry, they are very different. Now, this study was done at La Selva Biological Station. This is a very famous site for field biology and is located in the Caribbean slopes of Costa Rica. And this is part and managed for several years by the Organization of Tropical Studies, which is actually having its 60-year anniversary. So they've been celebrating about that. And the part that I'm going to talk to you about is essentially my first chapter of my dissertation, and it relates on how seed traits are related to a species reproductive performance. So this conceptual figure is mainly summarizing how seed traits are at the center of different trade-offs that plants have in order to produce their offspring. So we have two stages, the pre-dispersal and then the post-dispersal, when you actually get the seedling already merged. So my dissertation is focusing on how I'm seeing the relationships and the trade-offs between the seed and how the different aspects of plant reproduction, like phenology, reproductive output, their defenses, nutrients, and after the second part and a continuing part would relate to the seedling stage. Now, at the field, what I basically did was going along the station, along the trails, looking for Piper species and collecting vegetative trails. That means traits. That means the number of leaves, the percentage of herbivory, and then reproductive data. That means the number of fruits per plant, the number of seeds per fruit, and also phenological data that has been collected for a couple of years now. Now, at the same site at the lab, then I will start measuring leaf area, leaf toughness, leaf thickness. These are all traits that are usual for establishing different aspects of plant fitness. And in terms of reproductive traits, then I will focus on seed size and started doing germination trials. Now, all of this data is still being processed, and the first part that I'm going to show you about it's related to leaf functional traits. So in this graph, you can see at the bottom panel, we have the different species of Piper that we have. On the first, the bottom panel here, It's leaf area, and on the top panel is leaf thickness. So these traits are not much informative. So it means that we have different species that they don't differ that much in their thickness or in their size. And the graph here in different colors is showing you the levels of herbivory per species. So in this part, we don't see much difference that would relate to their functional traits. Now, relating to seed size, here is where we have a lot of variation. So, and these photos are showing you the different morphologies of fruits that we have, and the striking variation in seed size on the right, especially. So this picture was taken at a scale. So you can see that we have species with really tiny seeds, and then that go they grow a lot and they differ a lot. Now, so all this data was then summarized, and here is a graph that's showing you the seed area versus their dry weight. So this line is showing you what you would expect from a one-to-one relationship. That means if the seeds are as big as they weigh, they should fall along this dotted line. But as you can see, all these piper species are actually falling far from this one-to-one relationship. So here I want to point your attention to particular cases here, the one that looks more like an outlier. This one, it's way far from the one-to-one relationship. And the other point that I want to focus is on these two species here. So you can see that they are really close together. So if you think about the size and how they look like, these pictures are showing you how alike they are. So these two belong to two vine species, so they have similar growth habits. They grow in open spaces, so their environment is pretty much the same. And the idea of this chapter is, and what is currently on the works, is to look at the chemical diversity they have. So, sorry, this looks all moved out. So the idea, if you go back to this, to the phylogenetic tree that we have, showing where the species closely together are very similar phylogenetically, but they are really different in their chemistry. This is all based on the chemistry from the leaves. Now, what we want to see and what is not yet understood and not documented is the chemistry of the seeds. How is this actually translated into the differences between seeds, especially in cases like this, where you have plants with similar habits, similar environment, and similar size, but they are still coexisting together. Now, part of the work down there was also shown at a cultural and environmental fair done at La Selva, where we're doing activities like this not only focus on science, but also on how we can communicate it. So we don't need high tech to show these little works, little videos that unfortunately I can't show you right now. But this was really interesting for kids and adults that were at the station. So if you, let me see if I can play this. No. Okay, so at this fair, you can see this plant is one of, it belongs to Piper darien-ense. It just belongs to it's between Colombia and Panama, and some species reach Costa Rica. So this one is quite particular because when you chew the leaves, they feel like an anesthetic. So this is a species that they used to have it for controlling toothache, because it actually anesthetizes your mouth. So if you start chewing it, you won't be feeling anything for about 2-3 minutes. And that was very interesting for kids down there. So thanks to for the Tinker Foundation and this kind of work that we can do down there, we can also show it to the actual community, which I found that was pretty neat. Okay, and with that, I'll take any questions if you have them. Thank you.
You mentioned earlier you were looking for signs of herbivory. What kind of animals are eating these plants? Do they also get that anesthetizing effect from the seeds?
So that anesthetizing effect is just for one. This one I show you at the fair. Not all of them have that. They do have several interesting chemistry, but the main herbivores of these plants are caterpillars. And there is a neat relationship, how these caterpillars actually can detoxify in a way all the compounds that they chew from the leaves. And all these theories based on what we know about the chemistry of the leaves, but we don't know anything about the seeds. So this is completely new ground, and that is what actually you can stay tuned to find what happens afterwards.